for the introduction. Yeah, I still remember that last time when I presented at the ICP symposium is back in 2019. That's like 4 years ago at Hopkins. And now 4 years has passed, and I think the non-contrast, the BBB techniques also evolves a lot. So today I'm going to talk about the recent progresses in this field and also its clinical applications. So as we all know that the blood-brain barrier is a highly selective barrier formed by the endothelial cell tight junctions, the astrocyte and feet, parasite, basal lamina, and it will prevent the macromolecules and neurotoxins to go into the brain. So it is crucial for the normal brain function. And we all know that the BBB is disrupted in many diseases such as ALS, tumors, MS, Alzheimer's disease. So of course, it is important for us to measure the BBB permeability. Conventionally, people will use, for example, the lumbar puncture to measure the CSF serum albumin ratio, which means that you have to insert this needle into the spinal cord of a patient and get this albumin in the CSF. And this is a highly invasive procedure and will require very experienced physician to do this. And it is not feasible in a lot of large clinical studies. And the second, you can also use DCMRI to look at the post-contrast enhancement because the contrast agent will leak through this disrupted BBB. And for example, you can see this enhancement here in the tumor region. And if you scan a long enough time, for example, 20 to 30 minutes, then you can actually fit for this signal time course to a kinetic model so that you can get this K-trans, which is the transfer rate of the contrast agent through the BBB from blood into the tissue. But this kinetic model itself is very complex, and also the result is highly dependent on the model you are choosing. Also, there are a common problem of these two methods, that is either the albumin or the contrast agent we're using is large molecule, which means that they might be less sensitive to the early disruption of the BBB, especially at the early stage of the disease. So here, as said by the previous talk, we're trying to develop some new method to look at the BBB permeability to water molecule. And the main reasons are, first, the water molecule is a small molecule, so it can be more sensitive at the early stage. And the second is that the water is a main source of the MR signal, so it can act as an endogenous tracer. We don't have to inject any tracer anymore, so it can work as a non-contrast measurement. So like at very early days, people think that this water exchange across the BBB is totally free, but actually it is not. At around 1970s to 80s, there are several groups using very invasive methods to look at the BBB water exchange in either these animal models and also humans. And they found that this water extraction fraction by the tissue from the incoming blood is around 75 to 95%, which means that this BBB water exchange is indeed limited. And it can be limited by two layers. One is the endothelium. There are these endothelium cells and also tight junctions between them. So the water can go across the endothelium either by diffusion or filtration. And also after that, it has to go across the aquaporin 4 channel, which are the water channels, to go into this glial cell end bit and then go to the interstitial fluid and also the neurons. So by all these layers, the water exchange are limited. So that gives us the basis that we can actually measure that. So before I actually goes into those non-contrast BBB techniques, I still wanted to say a little bit about the contrast-based method, because the contrast-based MRI method can also measure the water permeability. It is not just for the contrast permeability. So the basic idea here is that if you inject the contrast agent into the vessel, into the blood, it will cause a decrease in the blood T1, of course. But because of this water exchange between the blood and also tissue, it will cause this apparent T1 of this entire voxel to decrease level is indeed dependent on the water exchange rate. So people are saying that this water exchange will push this system into a from the fast exchange regime to a finite or slow exchange regime, and they call it like a very fancy names. But overall, we can use some models to fit for this kinetic process. For example, you can measure the signals after the first pass of the bolus, and then use this shutter speed model to fit for this relationship between your R1 of tissue and R1 of blood. And this relationship is indeed dependent on the water exchange rate. Secondly, you can also use some new sequences such as a multi-flip angle, multi-echo sequence to give you more information, and that will characterize this two-compartment model better. And using these sequences, this group has also fit for this PS value, which is the BBB permeability index. It is called the permeability surface area product, and I will go to that in detail later. So essentially, they found that this PS value is directly related to the count of occluding, which is a protein that is responsible for the tight junction of the endothelium. So by this histological, histological examinations, you are saying that we can indeed use MRI to look at the BBB permeability. In addition, as we all know that the contrast agent method are often used to look at the cerebral blood volume, but the CBV estimation from the contrast MRI is actually biased by the water exchange. So for example, at different PS value, different permeability, your measured CBV is actually different than if you compare with the true values of CBV. So this bias itself can be used to look at or to fit for this BBB permeability. You can also derive some kind of index like this water exchange rate index. And it has been saying that in a stroke mice model, this contralateral, compared with the contralateral side, the stroke side indeed have a increase in the BBB permeability. In addition, I also want to say that recently it has been proposed that we can use the contrast to enhance the ASO, which means that you measure the ASO signal pre-contrast and post-contrast to a model fitting to get the permeability as well. But this actually belongs to the ASO-based method. I only mention it here because it requires a contrast injection. So that's for the contrast method. But we always know that the contrast has a lot of problems. It cannot be used in people who have kidney diseases. It has in the brain. It also invasive. So many groups have proposed several methods to look at the BBB permeability without using the contrast agent, and that goes to our non-contrast techniques. So most of the non-contrast techniques are ASL-based methods. So imagine that you label the water springs using ASL pulses at the artery site. It will go into the brain and exchange with the tissue. So the signal at this tissue voxel, or I mean not tissue voxel, at one certain voxel is dependent on a lot of parameters. First, it will depend on the blood flow, which is of course the fundamental of the ASL. And also because this water exchange between the intravascular space and the extravascular spaces, the signal is also dependent on the transfer rate, or this K of water, and also dependent on the cerebral blood volume, and of course the T1 tissue and T1 blood. So the basic idea here of all the ASL-based method is that you wanted to separate the labeled spins in the blood compartment and tissue compartment so that you can fit for this transfer rate. So the first thought would be that we just do a multi-delay ASL and fit for this very complex two-compartment model to get this PS value. However, this is not feasible because the SNR of AS is just so low, right? And this fitting is actually dependent on the T1 difference of the blood and the tissue, and that is actually pretty small. So you cannot just use a multi-delay ASL to fit for this thing. So many groups have proposed to separate the ASL signal at the two compartment by other different properties between these two compartments. So the first one would be the diffusion prepared ASL. That is, the water spins in the blood compartment and tissue compartment, they are different in their diffusion property. So you can just insert a diffusion preparation module before your ASL readout. And with this model, you will be able to calculate this water exchange rate. They call it KW. But of course, they need a ATT maps to calculate for this value. But this original version of the sequences is truly, it's 3D indeed, but it has very low resolution, and also it kind of gives you only the KW value, which is not only dependent on the permeability itself, but also dependent on the cerebral blood volume. So recently, they have proposed that we can do a multi-delay diffusion prepared ASL. So with more data, of course, we can fit for not only the transfer rate, but also the water extraction fraction and also the PS values. So they have also done some clinical applications using the diffusion ASL sequence, and they found that in normal aging, there is a decreased water exchange rate. And it is also decreased in the small vessel disease cancer patient. They're also saying that the decreased KW is related to more severe AD pathologies, especially the CSF A beta 42. And they explain this by a depolarization of the AQP4, the water channels in those diseases. However, this is a little bit indeed counterintuitive that we saw in those diseases, the BBB permeability is increased, but of course this is water, so we don't know what actually happened. And second, of course, in addition to diffusion, we can use a T2-based ASL, just like the previous talk. We can separate the intravascular and extravascular labeled spins by the different T2 of the two compartment. And this was originally proposed by Matthias Kunz's group. And then it was adapted by Jack Ross' group into the mice models. And they indeed found that in the AQP4 knockout mice, that means they don't have any water channels anymore, there is an increase in the exchange time. And that indeed makes sense, right? Because there is no water channel, so the water exchange time is increasing. And they also found that in the aged mice compared with the young or adult mice, there is this decrease in the exchange time. So based on this findings, in normal aging, they're saying this water exchange or water permeability is decreasing. And this has also been updated in humans with a Harman coding T2 preparation and more fancier sequence designs so that we can get a better unification of the water exchange. So besides T2 and diffusion, we can also use some other properties. For example, this ideal method relies on the different intrinsic diffusivity encoding of the two compartment when you use a segmented grid readout. So essentially, you can use a grid readout with a low segment and a higher segment number. So you will be able to calculate the extraction fraction and the PS value as well. And using this method, this group has also demonstrated a increase in the extraction fraction and a decrease in the PS value in the lesion errors in the MS patient. In addition, you can also use the difference in the empty effect between these two compartments. So because we know that the spins in the blood compartment has much less empty effect compared with the spins in the tissue compartment. So on the 70 scanner, this group have proposed that you can just insert a empty module here before your readout with the on and off together with SL. You will be able to get this maps of PS and also extraction fraction. Okay, I guess that's for all the ASL methods that's available now. But in addition to ASL, you can also label the water spins with diffusion blocks, not just the ASL pulses. You can label them with a diffusion block here. And this is called the FACSIG method, the future exchange imaging. And the basic idea here is that you first use a diffusion gradient to crush the spins in the artery, not in the artery, in the blood compartment, wait for a mixing time, and then use another detection block to detect your signal. And the signal decrease here is dependent on how fast the water exchange between the two compartments. And it has been found that this AX, or the exchange rate of the water across the BBB, is increased in the hippocampal region when you compare the MCI with normal controls. However, it has been argued that this vaccine model itself has still has some limitations, that it is highly dependent on the T1 relaxation, T2, and also this pressure gradient when you are doing all these diffusion modulations. So I guess there are still a lot of improvement has to be done in this vaccine signal model. So to summarize a little bit, that the common idea of all these non-contrast based methods are that you have to first use a labeled water spins as an endogenous tracer, and then you wait for that to be exchanged between the tissue and blood compartment. As long as you can separate the tissue compartment and blood compartment, you will be able to calculate this water exchange rate. However, all these methods, they indeed requires a fitting of a very complex two-compartment model. And because the ASO signal is indeed very low, we have to assume a lot of things, for example, CBV or ATT. Of course, you can also measure the ATT, but it may also be related to the oxygenation, etc. So it is indeed pretty complex process here. Another thought is that since all the labeled spins, they goes into the tissue, but not all of them are exchanged with the tissue. There will be some water spins that are labeled, but not goes into the tissue, and they will go directly to the venous side. So here, our method is that instead of measuring the signal at the tissue level, not just the, we don't want to fit all this very complex data modeling. We just want to measure those labeled spins at the venous side so that you can go back into this compartment model and then calculate for that extraction fraction of the water spins. So here is just the animation of the idea that I just talked about. Let's say if you label the water spins in the incoming artery, it goes into the brain and exchanged by the tissue. Most of those label spins are extracted, but still some are not extracted and goes directly into the venous side. So as long as we can measure those spins at the venous side, you can calculate how much of the water spins are extracted by the tissue, and that extraction fraction is directly related to the BBB permeability. And another illustration, essentially, we want to label it with, for example, Picasso, and then wait for a very long time. Here, we normally wait for like 3 seconds or even 4 seconds to wait for those spins to arrive at large veins. And here we choose a very large vein in the brain, that is the superior central sinus, which drains most of the spins from the cortex. And here, the signal at the superior central sinus will be dependent on, one, your water extraction fraction, that is what you're interested in. And also, it is dependent on the bolus arrival time, which, because there is a T1 decay of the signal. And if you are doing a multi-delay, for example, you will be able to fit for this extraction fraction, and together with a cerebral blood flow, you will be able to get this permeability surface area product, the PS value, that is the index of the BBB. So the original ASO, of course, it works for this subject, but for example, it doesn't really work for this subject. The reason is that the signal at the SSS, the superior central sinus, is just low, and it is at the similar level of the perfusion signal. There will be a lot of partial voluming if you do not deal with this problem. So here we propose that we can add a bipolar gradient after the excitation in this acquisition module, so that you can just separate the venous spins or venous signal and the tissue signal. And that is just the same idea as the phase contrast. So you can actually do a selective imaging of the venous labeled spins. For example, here you can see a very bright superior central sinus signal here, but not the labeled water spins in the tissue. And that's what we call the water extraction with phase contrast arterial spin tagging, or webcast MRI. And here are some representative results from the webcast. As you can see, we indeed do a multi-delay webcast, and we see the signal follows a connecting model. You can fit for that and get the extraction fraction and the bolus arrival time, and the extraction fraction gives you the PS value. However, we always know that the clinicians, they don't want a multi-daily ASL. It's just too long, right? It's not feasible in a lot of clinical studies. So that's why we designed another version that is a single delay version of webcast. So the only reason we're doing a multi-delay is because we want a bolus arrival time. But instead of asking what is the bolus arrival time of our signal, we actually utilize the fact that we're acquiring the signals along the entire sagittal sinus. We're doing a sagittal acquiring. And we're asking that along this entire sagittal sinus, where the location has a known bed. So we know that because of the dispersion of the bolus, the middle of this bolus will have a peak signal, and that correspond to a bolus arrival time of half of your labeling duration plus the post-labeling delay. So here we are transferring this temporal information into a spatial information, and then we're able to do a extraction fraction calculated from just a single delay webcast, and that is correlated with the multi-delay webcast. And that means we can get this not bolus arrival time, we can get this BBB permeability in just 5 minutes. So with this much shortage of this shortening of the scan time, we can do a lot of other studies, not just the clinical study, but also some technical verification. So first, we indeed implemented the sequences on both Philips and Siemens scanners, and we want to know whether it is reproducible. And of course, we saw that the COV of the sequences is around like 8%, which is similar as ASL. So we do have a good reproducibility. Second, we kind of validated our sequences with a contrast-based method. And just as I said before, the contrast-based method can also be used to measure the water permeability. Here we used the basal method, which was originally proposed to measure the CBV. And here we fit the basal signal after a contrast agent injection. And this fitting will give you the PS value as well. And we see that there is a good correlation between your basal PS and the webcast PS, suggesting that we can indeed use this non-contrast method to give you a BBB permeability. Another question here we want to answer is that whether our BBB measurement is indeed stable under different physiological status. Just as Daniel just said, our brain is always affected by the caffeine. And for example, you are doing a scan, you don't know if this patient just had a cup of coffee or whatever, and that may affect your BBB permeability measurement. So here we did 2 experiments. One is the CO2 inhalation, and second is the caffeine ingestion. We want to use these stimulators to sort of change your physiology and see if the BBB permeability measurement is still stable or not. So here we found that after CO2, there's an increase in the CBF, of course, a decrease in the bolus arrival time, and a decrease of the extraction fraction, but the PS value remains relatively stable. After caffeine ingestion, of course, there is a decrease in the CBF, a decrease in the venous oxygenation, CMR2, and an increase in the water extraction fraction. But again, the PS value remains relatively stable. So that means no matter what physiological status our patient or our subject is in, we can use this sequence to get a relatively stable BBB permeability measurement. So in addition to this normal physiological investigation, we also did a small studies to look at how the BBB permeability change after a single bout of aerobic exercise, because we know that the aerobic exercise is often used as an intervention or treatment strategies for different neurodegenerative diseases. So here we found out that after a 10-minute or 20 minutes of running, the young subject, they showed an increase in CBF, and that is similar to some longitudinal studies to look at the CBF changes after exercise. We also see an increase of the CMR2, especially for that 20-minute group. And for the BBB permeability, it's actually different than I thought. I thought that the BBB permeability shouldn't change after just a single bout of exercise. But here we found that there is an increase of the PS, and that may be related to the vasodilation because the blood pressure increases after exercise. And as we know that PS is called permeability surface area product. It is not only related to the permeability of a single area of vessel, it is also related to the vascular surface area as well. So if there is a vasodilation, yes, maybe the PS will increase. So after all this technical stuff, technical verifications, I guess it's a little bit tedious. So I can show you some other clinical applications of our techniques finally. So I guess similarly as the previous talk, people are always very interested in how BBB changes in cognitive impairment. And of course, the BBB dysfunction has been associated with Alzheimer's disease. For example, it has been seen that there is an early disruption of the BBB in the hippocampal region. But of course, this study has been done in a USC group, which are actually recently got some trouble. I don't know if these results are true or not. Yeah, I guess people know about that, right? But nevertheless, there is also another study saying that the amyloid deposition is related to the bacterial infection, but the bacteria can only go into your brain because there is a leakage of BBB, right? And maybe the BBB is not related to the amyloid, according to this study. So we don't know. It's just that there are so many controversies in this area, in this field. In addition, the BBB dysfunction is related to the vascular risk factors. For example, in areas with a white matter hyperintensity, we saw an increase in the BBB permeability. And there is also some studies associated this BBB dysfunction with the increased perivascular spaces. So our question here is that since the BBB dysfunction is related to both Alzheimer's disease and vascular pathology, is there any mechanistic differences in these two relationships? And if there is, how can we demonstrate that? So here our hypothesis is that the BBB dysfunction is related to both Alzheimer's disease and vascular pathology, but their relationship are on different spatial scales, and we can use tracers with different molecular size to demonstrate that. So first, we used our webcast MRI to look at the water permeability, and that represents a index for the small molecule permeability. And the second, we used the lumbar puncture to look at the albumin permeability, and that represents a large molecule. Here are our results. First, we saw that there is indeed a decrease of the webcast signal in the MCI patient compared with the controls, which means that their water extraction fraction is increased. And indeed, when you calculate this PS value, you will see that there is this increase of the permeability in the MCI patient compared with the controls, but not the albumin ratio. And we also saw that this PS value of water permeability is directly related to their memory scores and also the composite cognitive scores, suggesting that this BBB dysfunction is indeed an early event in the Alzheimer's disease. However, BBB permeability related to different pathology. So first, we look at the AD pathology, that is the CSF A beta 42 level and also the CSF p tau level. And we saw that the PS is increased in those who have a more severe AD pathology, which means that the water permeability is indeed related to the AD pathology, but that is only the water permeability. We didn't see any correlations with the albumin permeability. But we do see that people who have a higher vascular risk scores have a higher CSF serum albumin ratio, especially in people who have hypercholesterolemia, suggesting that the albumin permeability is more related to the vascular pathology. So combining all the findings that we have in this study, we actually proposed a potential model of the BBB disruption in cognitive impairment. That is, the BBB leakage to small molecule is more related to the amyloid or AD pathology, but the vascular risk factors is more to the large molecule. And both of them could cause a potential to better understand how actually the BBB works in Alzheimer's disease or cognitive impairment, our techniques has also been expanded into animal platform because animal models can help you to do a lot of longitudinal studies, to do a lot of different modelings. And of course, there are several differences in animal models compared with humans, but overall, we can still get a relatively good or stable measurement of the water permeability in those animal models. And here we did a longitudinal study in those AD model mice, and we found out that as early as 6 months, there is an increase in the water extraction direction when you compare the AD mice with controls. And also the PS started to increase significantly at 7 months. So what does that mean? The 6 months in the mice actually correspond to a 30 years old of humans, which suggesting that the BBB dysfunction or disruption is indeed a very early event in the Alzheimer's disease. We also used a diffusion-based method to look at the water exchange across the intercellular water exchange. That means that the water exchange is not only presented at the BBB level, it is also exchanged between the cells in your brain. And we indeed find out that the exchange rate or the exchange time of the water is indeed decreased in the AD mice, especially in the hippocampal region. That means there is increased water exchange across the cells in the brain in the AD mice. And which means that this coexisted with our BBB leakage. And we are actually asking that, is this actually related to the glymphatic system? Because we see that there is this entire water circulation system may be changed, but we don't actually have any CSF measure here. So we don't know whether it is related to glymphatic or not. Yeah, I guess that's almost all for the AD session or AD diseases. We also did a bunch of small studies to look at how the BBB changes in other diseases, for example, the very pediatric sickle cell diseases, because as we all know from the yesterday's talk, the sickle cell disease is an inherited genetic disease, and in those patients, you will have these sickled cells. And because these sickled red blood cells, they will attach to your endothelium, it will cause an inflammation of the endothelium. And we all know that this endothelium is a most important component of the BBB. So we're wondering that if the BBB disruption is one of the problem or contributing factor in those diseases that cause a future stroke or cognitive impairment or whatever consequences. So here, using the non-contrast method, we indeed see that there is this increase of the PS values in the SCD patient compared with their siblings, the SCT, kind of like a control. And this PS increases is also directly associated with their hemoglobin level and also the hemoglobin S level, suggesting that there is indeed a BBB disruption in the pediatric sickle cell diseases. And it could be that there are some relationship with the cognitive function, but due to a small sample size, we really didn't. In addition, oh, sorry. Yeah, okay. I guess people are hungry now. Just yeah, briefly mentioned, we also did that in the severe COVID, we saw an increase in the BBB permeability, but to be sure that there is no BBB disruption in the Omicron infection, we did that in like 100 or 150 young healthy volunteers. So I guess with Omicron infection, we're good. And also the BBB disruption is presented in the demyelinated diseases, and it indeed helps us to differentiate the NMOSD, which is a neuromyelitis optica spectral diseases, with MS, because these are two demyelinated diseases which are often misdiagnosed. So we saw there is an increase in the PS in NMOSD, but the OEF actually decreases in the MS. So it is still an ongoing project. Yeah, and we also did Huntington disease mice, and the interesting thing is here we also do a 0-1 and Claudin 5 to suggest that this BBB disruption is indeed related to the tight junction. Yet to summarize very quickly, BBB permeability indeed can be measured with non-contrast MRI techniques, and a lot of studies have shown that this is a very valuable index in a bunch of diseases, and hopefully there are some more interesting studies in the coming years. And thank you for joining my Oxford Institute, and thank you.